Hello students, let us start with the next part of the photosynthesis. There are certain factors which are found in the external environment followed by within the plant what can be named as the internal factors which affect the rate of photosynthesis. So among the external factors, there are 4 different factors which affect the rate of photosynthesis, such as intensity of light, amount of carbon dioxide, or what do we call it as a carbon dioxide concentration, and temperature and water content in the soil. Now let us start with the first 2 factors. How do they affect the rate of photosynthesis, which can be represented in a graph? Children, these 2 graphs have been named as A and B. So when you just observe the first graph, you can observe that there is an increase in the intensity of a sunlight, and when there is an increase in the intensity of a sunlight, the rate of photosynthesis increases, and at this intensity of sunlight, the amount of carbon dioxide in the atmosphere is around 0.02%. So the rate of photosynthesis increases gradually, but after certain period of a time, you can find that the photosynthesis becomes constant, or you can say it is stabilized, or you can say it reaches a stability which can be named as a S1, right? So in case if there is an increase in the amount of a carbon dioxide from 0.02% to 0.05%, and again if there is a high intensity of a sunlight, again there is an increase in the rate of photosynthesis from S1 to higher level, and after reaching certain position, again it becomes constant, or you can say it stabilizes, which can be named as S2. So thus you can say that the rate of photosynthesis increases with increase in percentage of carbon dioxide in the atmosphere, even though there is a high intensity of a sunlight. But after a certain period of a time, even that rate of photosynthesis stabilizes, which you can learn more clearly when I teach you the later part. Fine. Now let us study about the temperature which affects the rate of photosynthesis. Children, the temperature required for the maximum rate of photosynthesis to occur is at around 35 degrees Celsius, which can be named as a optimum temperature required. So it means up to 35 degrees Celsius. So when there is an increase in the temperature up to 35 degrees Celsius, you will find that there is an increase in the rate of photosynthesis. In case if the temperature is around 22 degrees Celsius around 10 o'clock in the morning, by 12 o'clock if the temperature rises up to 32 degrees Celsius, so it means you can observe that there is a change in the temperature by 10 degrees Celsius. So right from 9 o'clock to 12 o'clock. So in that condition, you can say that the rate of photosynthesis doubles. Or if there is a temperature around 10 degrees Celsius and increases by another 10 degrees Celsius, reaches 20 degrees Celsius, so you can say that again there is an increase in temperature by 10 degrees Celsius. Even in that condition, you will find that the rate of photosynthesis doubles up. And remember, this doubling can occur only up to 35 degrees Celsius. But after 35 degrees Celsius, when the temperature reaches 36 degrees Celsius, 37 degrees Celsius, you can find that there is a gradual fall, or you can say there is a decrease in the rate of photosynthesis, because higher the temperature, the enzymes which are taking part in the photosynthesis, especially in the dark reaction, there is one particular enzyme known as a rubisco, which actually takes part in the dark reaction. That particular enzyme will be destroyed and it stops at 40 degrees Celsius. So you can say that higher the temperature beyond 35 degrees Celsius, the rate of photosynthesis reduces and stops at 40 degrees Celsius because the enzymes are destroyed at a high temperature. Fine. Moving on to the next factor, that is water content. In case if there is a less absorption of water by roots, or if there is a high transpiration, or the water lost by transpiration is very high, so this leads to scarcity of water in the plants. So when there is a scarcity of water in the plants, immediately the stomata are closed. So when the stomata are closed, there is no inward diffusion of carbon dioxide, thereby the photosynthesis reduces. Fine. So though the plants absorb so much of water, only around 1% of water is utilized to carry out photosynthesis. Fine. So thus these are the 4 external factors which affect the rate of photosynthesis. So what is the basis on which you can say that the rate of photosynthesis depends on? Now let me give you one such example. Let us consider 4 days in a week. Let me name it as Monday, Tuesday, Wednesday, followed by Thursday. Let us provide all the external factors to a particular plant. Let us imagine that all these factors are provided to the plants, such as intensity of a sunlight, carbon dioxide concentration, temperature and water content. Let us start. What happens on Monday? So Monday there is a more intensity of a sunlight. There is 0.03% of carbon dioxide, which is a suitable amount to carry out photosynthesis, and the temperature is around just 10 degrees Celsius and plant has a sufficient water. But can you just guess what happens? Can you just look at the factor which actually affects the rate of photosynthesis? You can observe that there is one factor which is very less, that is the temperature, which is just 10 degrees Celsius, which will reduce the rate of photosynthesis on Monday. So if there is an increase in the temperature, of course, even the rate of photosynthesis increases on Monday. So let us move on to the second day, that is Tuesday. On the second day, there is a cloudy day. So it means there is a less intensity of a sunlight. So there is a less intensity of sunlight, and we know that when there is a less intensity of a sunlight, the stomata are partially closed. Fine. Of course, there is a suitable amount of carbon dioxide, and 31 degrees Celsius is a temperature which is very close to the 35 degrees Celsius, which is the optimum temperature, and there is a sufficient water content in the plant. Fine. But again, the rate of photosynthesis on the second day is also affected because there is no proper intensity of a sunlight, because it is a cloudy day, which will affect the opening of stomata. So if there is no cloudy day, again you can say that the rate of photosynthesis increases even on Tuesday. Moving on to Wednesday. Again on Wednesday there is a sufficient intensity of a sunlight, but look at the amount of a carbon dioxide. It is around 0.01%. Temperature is 30 degrees Celsius, a very suitable temperature, and water content is sufficient. But since there is a less concentration of a carbon dioxide on Wednesday, again the rate of photosynthesis is affected even on Wednesday. If there is a slight increase on Wednesday with respect to carbon dioxide, of course the rate of photosynthesis will be doubled up, I can say. Then moving on to Thursday, there is sufficient intensity of sunlight, sufficient amount of concentration of carbon dioxide in the atmosphere. Temperature is 33 degrees Celsius, but look at the water content. The amount of water content is very less in the plant. Okay, so just now I said that if there is a less amount of water in the plant, or if there is a less absorption of water by the roots and more transpiration, automatically the stomata closes and inward diffusion of carbon dioxide does not occur, which will reduce the rate of photosynthesis. So basically, if you just look at the different days of this particular week, you can very clearly say that, Monday the rate of photosynthesis was less because of temperature, which was too less. If you increase that, the rate of photosynthesis will be more. So in the same way, Tuesday it was a sunlight, Wednesday the amount of carbon dioxide, Thursday the water content. So you can say that in the nature you will never find all the conditions being favorable to the plants. So you will have one or the other factor which is in shortage, which is in limited supply. So you can say that the rate of photosynthesis depends on the factor which is in short supply. So finally you can say that the rate of photosynthesis depends on the factor which is a short supply, which is in short supply. Okay, so this we call it as a Blackman's principle, which is not in the syllabus, but he was the one who stated that. So the rate of photosynthesis always depends on the factor which is in short supply. If that factor is increased, the rate of photosynthesis doubles up or increases. Fine, children. Now let us move on to the internal factors which affect the rate of photosynthesis, which are found within the plants. Fine. If you remember, chlorophyll comprises carbon, hydrogen, oxygen, nitrogen and magnesium. So these are the elements that make up the chlorophyll, which is very much essential to absorb sunlight to carry out photosynthesis. In case if there is a nutritional deficiency, Okay, if there is a lack of nutrients provided to the plants, in that way there is no formation of a chlorophyll, which will affect the absorption of sunlight and the rate of photosynthesis reduces. Then moving on to the second internal factor, protoplasm. So what is the meaning of a protoplasm? Protoplasm is nothing but the nucleus along with the cytoplasm is known as a protoplasm. Okay, so nucleus along with the cytoplasm makes the protoplasm. In case if there is a dehydration, if there is a lack of water in these cells or in the protoplasm, definitely that will affect the photolysis of water and the rate of photosynthesis will be very less. If not dehydration, in case if there is a accumulation of starch, we know that the photosynthesis, there is a formation of a glucose molecules and many glucose molecules will change into starch. And if this starch is accumulated in the cytoplasm of the cell without being translocated to the other parts of the plant, so this accumulation of a starch in the cell also affects the rate of photosynthesis on the following day. Right? So this also can be one of the internal factor, that is dehydration of protoplasm or accumulation of a starch in the cell will definitely affect the rate of photosynthesis. And then moving on to the structure of a leaf, I think you can guess, children, if there is a thick cuticle, okay, the cuticle must be transparent. If the cuticle is very thick, so the absorption of sunlight is affected, which reduces the rate of photosynthesis. And if there are only few stomata, so again in that case there is a reduction in the inward diffusion of carbon dioxide for the photosynthesis. Okay, and then if there is a narrow, if you find the narrow leaves, okay, the size of the leaf is too small, so that maximum absorption of sunlight cannot take place, which also affects the rate of photosynthesis. Fine, children. So these are some of the internal factors which affect the rate of photosynthesis. Okay, now let us study about certain experiments which are related with photosynthesis. Right, now when we learn about the experiments in this chapter, we have certain common steps followed in all the experiments. Basically, what are the experiments in this plant that we are learning? In this chapter, we are going to prove that plants need carbon dioxide to carry out photosynthesis. Plants need sunlight to carry out photosynthesis. Plants need chlorophyll to carry out photosynthesis. So all these factors will be proved. Okay, but when you prove this, you're going to have some common steps which I'm going to teach you, okay, which is holding good for all the experiments. So what do we call that common steps? As we call it as a testing a leaf for starch. Okay, so to begin with, what is the first step we are going to take up when you are going to perform these experiments? The first step is we are going to destarch the plant. So within the word you have a clue. What is the meaning of a destarching the plant? It means removal of a starch. From where to where? You're going to remove the starch from leaves to the other parts of the plant, so that you want the leaves to be free from starch, so that on the following day you're going to expose this plant to the sunlight, and then you have a fresh glucose synthesized which is converted into starch, and then you're going to perform the starch test. So what you're going to do to destarch the plant, to remove the starch from leaves to the other parts of the plant, you're going to keep that plant in the dark room or in the cupboard for about 24 to 48 hours. And remember children, whenever it is at night, we know that all the starch found in the leaf changes into glucose and it is translocated to other parts of the plant that we are familiar. So you're going to provide that condition in the cupboard or in the dark room, okay, in the plant where you're going to conduct the experiment. So what is the meaning of a destarching plant? Destarching the plant means it is the removal of starch, very clearly, removal of starch from, you should say, from leaves to the storage parts of the plant. What is the meaning of storage parts of the plant? It is nothing but roots followed by stem. Or in a simple word, you can say that making the leaves free from starch. Fine children. So once you destarch the plant, after about 1 or 2 days, take that plant and keep in the sunlight in order to allow the plant to carry out photosynthesis. Right? So keep the plant in the sunlight for about few hours, and after few hours pluck one of the leaf. Just pluck one of the leaf and then you're going to test that leaf for a starch. Why not glucose, children? Because immediately after photosynthesis, a number of glucose molecules undergo polymerization and then they form the starch. So you can only find the starch in the leaves but not the glucose. Hence we are going to perform the test on the starch. Fine. So what you're going to do, pluck one of the leaf, take a test tube, fill the test tube with water and then place this leaf, boil the water. When you boil the water, it means we are going to boil the leaves. Why is that we are boiling the leaf? In order to kill the cells. Why should we kill the cells? Only when you kill the cells are nothing but those palisade parenchyma cells, spongy parenchyma cells, they have the starch inside them. So before you test the leaf for a starch, you should make the cells to be killed. So that is the reason we are going to boil the leaf in water to kill the cells. Okay, most of the time I have seen children, when I ask you this question, they just write it as killing the germs. Children, not germs, it is killing the cells of the leaf. Then after that, the second step is you're going to take that leaf in one more test tube and take the alcohol in that test tube, and in that alcohol you're going to keep this leaf, right? And you should be careful when you're boiling the alcohol. If you directly hold the test tube onto the Bunsen burner, the flame of the Bunsen burner, you know that alcohol is a combustible substance. If this alcohol spills out, okay, there are chances that alcohol may fall on the flame, which may lead to accidents. So in order to prevent that, we have to boil the alcohol indirectly by keeping the test tube in the beaker containing water in order to prevent the accidents that may occur if you are a little careless, right? So what type of alcohol you can use? You can use methyl alcohol, you can use ethyl alcohol, and then remember, this alcohol is a colorless solution, right? So after you boil this leaf, the colorless alcohol now changes into green color solution. Can you guess why does it change into green color solution? Because the chlorophyll will be removed. So the leaf will lose the chlorophyll, which makes the solution to turn green in color. So why do we boil the leaf in the alcohol? If they ask you, you should be very clear. You should say that in order to remove the chlorophyll. Okay, so once the chlorophyll is removed in the leaf will turn pale white. It changes into a pale white. Okay, and that pale white leaf has to be taken. You can again wash it in water to make it soft, and once you have that pale white leaf, it is ready for testing a leaf for starch. Okay, so what do we add to test the starch? We are going to add a iodine solution. Children, the chemical is iodine solution. You have to write it as a solution but not iodine, because iodine is found in the form of a solid. So that iodine is dissolved in the water and we get a solution. That solution we are going to use. So in case if they ask you what is a chemical used to test the presence of a starch in a leaf, it has to be iodine solution, right? So when you add the iodine solution, let me tell you, the color of iodine solution is brown in color. Right? So when you add the iodine solution on the leaf, if there is no starch in that particular part of a leaf, what happens? It remains brown. So what is that observation you are going to write? You are going to write that observation. In case if they ask you state your observation in that particular part of a leaf, you are going to write it as remains brown. Can you tell me why does it remain brown? Due to the absence of starch. So this you are going to write very clearly. And if there is a starch, what happens? If there is a starch, you may find the leaf turning into blue-black, or you can say bluish-black or a blue-black. So what does that blue-black indicates? It indicates the presence of starch. So remember, when they ask you about the observation of a leaf, you have to write these 4 words. Remains brown due to the absence of starch, turns blue-black due to the presence of starch. So for any experiment, you're going to use these 4 keywords to explain the observation. Okay? Yes, children, let me quickly tell you the steps followed. First, you're going to destarch a plant by keeping the plant in the dark room for about 24 to 48 hours, during which the starch from the leaf is removed and translocated to the roots or a stem. Then you're going to keep the plant in the sunlight for about few hours. After that, pluck a leaf and boil the leaf in water in order to kill cells. And again, transfer that leaf into a test tube containing alcohol, boil it in the alcohol, the leaf turns pale white because the chlorophyll is removed due to boiling the leaf in the alcohol. Then take that leaf, and the leaf has to be washed in the warm water, and then it is ready for the test. Then you're going to add iodine solution. So wherever you find the color is changing to blue back, indicates the presence of starch. Wherever you find if it remains brown, it shows the absence of starch. Yes, children. So these are the common steps for all the experiments which I will discuss along with your textbook. Fine. Moving on to the next experiment. Okay, so this is an experiment to demonstrate that oxygen is liberated. Oxygen is liberated during photosynthesis. And this is one of the diagram usually asked in the examination which you have to practice. So let us start with this experiment. I think children, you can try this experiment all by yourself at home. Let us see. So you are going to take a funnel, and if you look at the funnel has two parts. So we call this as a mouth of the funnel, followed by a tube-like structure. What do we call it? As a stem of the funnel. So within this mouth of the funnel, you're going to keep few branches of the aquatic plants. It can be hydrilla, you can take elodia. So the aquatic plants can be taken and kept inside this mouth of the funnel, and invert that funnel in a beaker. Then fill the water in the beaker. So fill the water in the beaker, and then take a test tube and fill water in the entire test tube up to the topmost position completely. It has to be filled with the water, and then invert it on the stem of the funnel. And you have to take a precaution that the stem of the funnel should be below the level of water in the beaker. Okay, even when you draw the diagram, you have to be very careful. The stem of the funnel should be below the level of water in the beaker. Fine. So you can keep this plant in the sunlight for about few hours, and then since we have taken the aquatic plant, we have taken the tap water or a fresh water in the beaker, you can hasten the process. You can make the process to occur quickly. How? Because plants require carbon dioxide to carry out photosynthesis. So you can add a pinch of sodium bicarbonate. You can add a pinch of sodium bicarbonate, that is baking soda, right? Baking soda into the beaker. So when we add the pinch of sodium bicarbonate to the beaker containing water, that chemical will liberate lot of carbon dioxide, which is a source for the hydrilla plant to carry out photosynthesis. So after some time, we find that bubbles of water start collecting on the surface of hydrilla, starts escaping through the stem of the funnel, and finally starts occupying the space in the test tube by pushing the water downward. So you can say that there is a downward displacement of water after some time. So in order to check whether is it oxygen or some other gas liberated, you're going to take the test tube and introduce a glowing splinter. So when you introduce a glowing splinter, it rekindles, which indicates that oxygen, which is a supporter of combustion, makes the splinter to burn into flame. So this clearly indicates that it is the oxygen which is liberated during photosynthesis. And children, this diagram has to be practiced. Okay, so with this we complete with the experiments, right? And then we'll discuss about the carbon cycle, when you just have your textbook along with you. Fine children, thank you.